This week we're going to be looking at the human brain, specifically focused on vision. So, introduction. In this lesson, we'll be exploring the human eye and how it works, as well as some of the different parts of your eye. Focus question. What causes color blindness? Is it your eyes or how your brain interprets what is being seen? So, you have 3 minutes to go ahead and answer that focus question on the Google form. All right. Seems like our timer has ended. As usual, if you need more time, feel free to pause the video and then pick it up when you're ready. All right, so we have a beautiful quote here. If the human brain were so simple that we could understand it, we would be so simple that we couldn't. That's by Emerson M. Pugh, I believe. So we have a color blindness. We're going to take this quiz, and I'll let you guys go to that, and then we're going to watch this video. The quiz uh, has you look at these different patterns, and within them they have numbers. And if you're able to see all the numbers, it shows that you have good enough vision to not have color blindness. Whereas some people might not be able to make out all of the different numbers, and that's because of some minor color blindness within their vision. And what we have here for the video is the first time reaction to enruchima, I believe is how it's pronounced, glasses, which enable people who have color blindness to be able to see. Warning, it is a little bit emotional. And if we can watch it.
Like this looks very at your kid's eyes, Petey. They're so pretty.
You can tell orange. What else is it? The streamers. Wait a second. Ah! It's green. I thought it was all orange. How did they think it was all orange? That's
Oh my god! Oh my god, they were cool. Oh my god. Oh my god, it's really beautiful.
You're supposed to keep them on. I know, but I have to compare some. Like, you can't tell me not to compare.
Do you know what color this is?
It's green. It's super green. Oh my god, it's so real. It is real. It is real. The cow. Look at the cow, man. This is spectacular. I can't even imagine how different the world looks. I swear to God. Like, it's so crazy. Purple.
You have purple in your shirt.
I do.
Purple and green.
I never knew that. I always thought the shirt was yellow and blue.
Dang.
All right. So, the human brain overview. Each part of the brain can be broken into different parts, which we call lobes, that all serve a different purpose, much like how each cell structure has a specific purpose within the cell. The frontal lobe is the part of the brain that controls important cognitive skills such as emotional expression, problem solving, memory, and language. The parietal lobe functions in processing sensory information such as location of body parts, as well as interpreting visual information and processing language and mathematics. The occipital lobe processes and interprets everything we see, such as shapes, colors, and movement. And the temporal lobe is the part of the brain that is involved in hearing and processing information into meaningful units, such as speech and words. So since we're looking at vision, we're going to be looking at the occipital lobe. Vision and how it works. So we have this great crash course video we're going to go ahead and watch.
Take a good long look at this. We're gonna mess with your brain. This is the first stage of
So
an
go ahead and
optical
stare
illusion.
at that
Many
as much as you
illusions
can.
use patterns of light or perspective to exploit the disconnect that exists between sensation and perception, between what your eyes see and what your brain understands. But not all illusions work that way. Some produce ghost effects or after images that take advantage of glitches in the physiology of human vision, like this flag. I'm not trying to make a political statement here, and I'm not gonna ask you to swear allegiance to the Republic of Hank or anything. I mean, if I was gonna start my own country, my flag would be way cooler than that. Not that I've thought about that a lot. And now look at this white screen. If you looked at that flag for at least 30 seconds without moving your eyes, you'll see something, even though the screen is blank. An after image of the flag. But instead of being turquoise and black and yellow, it's red, white, and blue. Okay, so that's pretty cool, but I'm not here just to entertain you. This kind of illusion is actually a great way to explain your very complex sense of vision. And I do mean complex. Nearly 70% of all the sensory receptors in your whole body are in the eyes. Not only that, but in order for you to see, perceive, and recognize something, whether it's a flag or a handsome guy in glasses and a sport coat sitting behind a desk, nearly half of your entire cerebral cortex has to get involved. Vision is considered the dominant sense of humans, and while we can get along without it, and it can be tricked, what you are about to learn is not an illusion. When we talked about your sense of hearing, we began with the mechanics of sound. So before we get to how your eyeballs work, it makes sense to talk about what they're actually seeing. Light bouncing off of stuff. Light is electromagnetic radiation traveling in waves. Remember how the pitch and loudness of a sound is determined by the frequency and amplitude of its wave? Well, it's kind of similar with light, except that the frequency of a light wave determines its hue, while the amplitude relates to its brightness. We register short waves at high frequencies as bluish colors, while long, low frequencies look reddish to us. Meanwhile, that red might appear dull and muted if the wave is moving at a lower amplitude, but super bright if the wave has greater amplitude and thus higher intensity. But the visible light we're able to see is only a tiny chunk of the full electromagnetic spectrum, which ranges from short gamma and X-rays all the way to long radio waves. Just as the ear's mechanoreceptors or the tongue's chemoreceptors convert sounds and chemicals into action potentials, so too do your eye's photoreceptors convert light energy into nerve impulses that the brain can understand. To figure out how all this works, let's start with understanding some eye anatomy. Some of the first things you'll notice around your average pair of eyes are all the outer accessories, like the eyebrows that help keep the sweat away if you forgot your headband at racquetball, and the super sensitive eyelashes that trigger reflexive blinking, like if you're on a sandy beach in a windstorm. These features, along with the eyelids and tear-producing lacrimal apparatus, are there to help protect your fragile eyeballs. The eyeball itself is irregularly spherical with an adult diameter of about 2.5 centimeters. It's essentially hollow, full of fluids that help it keep its shape, and you can really only see about the anterior sixth of the whole ball. The rest of it is tucked into a pocket of protective fat, tethered down by 6 strap-like extrinsic eye muscles and jammed into the bony orbit of your skull. While all this gear generally does a fantastic job of keeping your eyeballs inside of your head, which is good, on very rare occasions, perhaps after head trauma, or even a really intense sneeze, those suckers can pop right out, a condition called globe luxation, which you really do not want to Google. Sit here while you Google it. Now, you don't need to pop your eyeball out in order to understand how it's structured. I'll save you the trouble and tell you that its wall is made up of 3 distinct layers: the fibrous, vascular, and inner layers. The outermost fibrous layer is made up of connective tissue. Most of it is that white stuff called the sclera, while the most anterior part is the transparent cornea. The cornea is like the window that lets light into the eye, and if you've ever experienced the excruciating pain of a scratched one, you know how terrible it can be to damage something so loaded with pain receptors. Going down a little deeper, the wall's middle vascular layer contains the posterior choroid, a membrane that supplies all of the layers with blood. In the anterior, there's also the ciliary body, a ring of muscle tissue that surrounds the lens, but the most famous part of this middle layer is the iris. The iris is that distinctive colored part of the eye that is uniquely yours. It's made up of smooth muscle tissue shaped like a flattened donut and sandwiched between the cornea and the lens. Those circular sphincter muscles, yeah, that's right, you got sphincters everywhere, contract and expand, changing the size of the dark dot of your pupil. The pupil itself is just the opening in the iris that allows light to travel into the eye. You can see how an iris protects the eye from taking too much light in. If you shine a flashlight in your friend's eye in a dark room, their pupils will go from dilated to pinpoints in a couple of seconds. Light comes in through the cornea and pupil and hits the lens, the convex transparent disc that focuses that light and projects it onto the retina, which makes up the inner layer of the back of the eyeball. Your retinas are loaded with millions of photoreceptors which do the crucial work of converting light energy into the electrical signals that your brain will receive. These receptor cells come in 2 flavors, rods and cones, which I'll come back to in a minute. But the retina itself has 2 layers: the outer pigmented layer that helps absorb light so it doesn't scatter around the eyeball, and the inner neural layer. And this layer, as the name indicates, contains neurons. Not only the photoreceptors, but also bipolar neurons and ganglion neurons. These 2 kinds of nerve cells combine to produce a sort of pathway for light, or at least data about light. Bipolar neurons have synapses at both ends, forming a kind of bridge. On one end, it synapses with a photoreceptor, and at the other, it synapses with a ganglionic neuron, which goes on to form the optic nerve. So say you've just been hit with a blinding flashlight beam. That light hits your posterior retina and spreads from the photoreceptors to the bipolar cells just beneath them to the innermost ganglion cells, where they then generate action potentials. The axons of all those ganglion cells weave together to create the thick, ropey optic nerve, your second cranial nerve, which leaves the back of your eyeball and carries those impulses up to the thalamus and then onto the brain's visual cortex. So that's the basic anatomy and event sequencing of human vision. But what I really want to talk about are those 2 types of photoreceptors, your rods and your cones. Cones sit near the retina's center and they detect fine detail and color. They can be divided into red, green, and blue sensitive types based on how they respond to different types of light. But they're not very sensitive, and they really only hit their activation thresholds in bright conditions. Rods, on the other hand, are more numerous and more light sensitive, but they can't pick up real color. Instead, they only register a grayscale of black and white. They hang out around the edges of your retinas and rule your peripheral vision. Since these receptors function so differently, you might not be surprised to learn that your rods and your cones are also wired to your retinas in different ways. As many as 100 different rods may connect to a single ganglion cell, but because they all send their information to the ganglion at once, the brain can't tell which individual rods have been activated. That's why they're not very good at providing detailed images. All they can really do is give you information about objects' general shape or whether it's light or dark. Each cone, by contrast, gets its own personal ganglion cell to hook up with, which allows for very detailed color vision, at least if conditions are bright enough. And all this brings us back to that weird flag. Why does staring at this flag and then looking at an empty white space make us see a phantom flag of different colors? Well, it begins with the fact that our photoreceptors can make us see after images. Some stimuli, like really brilliant colors or really bright lights, are so strong that our photoreceptors will continue firing action potentials even after we close our eyes or look away. The other part of the illusion has to do with another bug in our visual programming, and it's just that our cones can get tired. If you stare long enough at a brightly colored image, your cones will receive the same stimulus for too long, basically stop responding. In the case of the flag, you looked at an image with bright turquoise stripes. Because your retinas contain green, red, and blue sensitive cones, the blue and green ones got tired after a while, leaving only the red cones left to fire. Then you looked at a white screen. That white light included all of the colors and wavelengths of visible light, so your eyes were still receiving red, green, and blue light, but only the red cones were able to respond. As a result, when the after image began to appear, those stripes looked red. The same thing happened to your rods, except since they only register black and white, the after image was like looking at a negative of a photograph. Dark replaced with light. That's how those black stars and stripes turned white. So yes, human vision is fallible, but those mistakes that it makes can help us understand that wonderfully complex system. And that wonderful...
And that's where it ends. Sorry. All right. So when you are seeing, you are taking in a variety of frequencies and amplitudes of light that bounce off the object you're looking at. So you're not really seeing them directly, you're seeing what's reflected off of them. Once that light is reflected, it then passes through different parts of your eyes. It starts with the cornea, then the pupil, iris, lens, retina, and then to the optic nerve. Before the optic nerve transfers that image to your occipital lobe to be processed. Within your eyes, you have rods which only register the grayscale of black and white, and cones which detect red, green, and blue colors. So we're going to be looking at the parts of the human eye. You can see there's a picture with various different parts. We're not going to talk about all of them. We are going to hit on some of the bigger, more important ones, not to say that they're not all important, just some of the large ones that we want to talk about. So the cornea is a transparent dome, and it helps the eye to focus light. And the cornea is right here. It's on the outermost edge of your eye. And the iris is the colorful part of your eye and expands or retracts to control the amount of light going through the pupil. So whenever you talk about somebody's eye color, what you're really talking about is the color of their iris. The lens's job is to focus the light rays on the back of the eyeball. And the lens is actually, we used to do the cow eyeball dissection. And the lens is actually it's kind of like those green stones we handed out in the beginning of the year. It's just kind of the same shape, same size. And all it does is just like the lens of a magnifying glass. It focuses the light and it helps to kind of enhance and enlarge the image just a little bit. And the retina is in the back of the eye and has millions of cells that are sensitive to light. The retina takes the light the eye receives and changes it into nerve signals so the brain can understand what the eye is seeing. The retina is right back here. It's that layer inside. And the optic nerve serves as a high-speed telephone line connecting the eye to the brain. When you see an image, your eye telephones your brain with a report on what you are seeing so the brain can translate that report and basically be able to make sense out of what you are seeing. One of the interesting things that we don't talk about in this video this lesson, is we used to do the cow eyeball dissection, and when we did that, I actually learned that when light comes in, the lens actually inverts it, so it flips it upside down. So whatever you're seeing comes into your eye flipped upside down, and then the retina and everything behind it, all of those cells and nerve cells are responsible for flipping that image and translating that to the brain. I found that pretty interesting when I was going through. So color blindness. So what causes color blindness? Usually color blindness is caused by genetics you inherit from your parents, and that is what causes color blindness. Sometimes it can be caused by damage to the eye, either physical or chemical. So for instance, you might have an injury where you have something go in your eye, you have like major trauma, or chemically, like when we talked about lab safety, that's why we wear goggles. We don't want to injure our eye to either go completely blind or even have color blindness. So those are some causes. So is it your brain that impacts color blindness, or is it your eyes? So in your eyes, you have three different kinds of cones that absorb various wavelengths of light, and each kind reacts to either red, green, or blue. These cones send information to the brain that distinguishes colors. So if one or more of these cones in your retina is damaged or is not present, you will have difficulty seeing colors properly. So you might just have some minor damage with your cones in your eyes, or some people are born where a certain type of cone is completely not there. So they don't see those colors at all. There doesn't react. So the focus question. What causes color blindness? Is it your eyes or how your brain interprets what's being seen? So let's go ahead and open up the Lesson 5 Google form and take 2 to 3 minutes to type in your thoughts. All right, and that is the timer. Again, if you need more time, feel free to pause the video and then continue when you're done. This has wrapped up this lesson. There are 2 quick things I would, if you're interested in this subject, I would advise you maybe look up on YouTube or learn a little bit more about. First would be, it's pretty fun to do, looking up on YouTube simple optical illusions or even after images. After images are exactly what we looked at the beginning of this video. It's like the flag where you have your overworking parts of your eye, specifically the cones, to where it'll flash to a blank screen or a flash to a black and white image, and you'll see color, even though there is no color there. The second one, and this might not be for everybody, I understand, is cow eyeball dissections. We did this my first year of teaching. It was, in my opinion, and a lot of students, probably one of their favorite labs. We got to get cow eyeballs, cut the fat off, open them up, and look inside. I would go ahead and you can find those videos on YouTube. They are very interesting to see. The reason that we would dissect cow eyeballs is because they are very similar to human eyeballs. So that might be something, if you're interested in, to look it up. It's very interesting, it's very cool, and I would highly recommend it. All right, well, have a wonderful week, and I will send out another lesson next week. I hope you all are doing well, staying safe, and enjoying yourself. All right, bye-bye.